All right, so we are on chapter 21, Patterns of Inheritance, part 1. And the main things that you should be able to do after this is contrast genes, alleles, genotype, and phenotype, contrast dominant and recessive alleles, determine the genotypic and phenotypic ratios within a monohybrid cross using Punnett squares. That's a lot of vocabulary, so make sure you know that. And determine the phenotypic ratio of a dihybrid cross using Punnett squares. So our question of the day is, what are the chances of a child with cystic fibrosis? And you don't necessarily have to know what this is. It's sometimes abbreviated as CF, but it's when there is an enzyme that is not functioning properly and it produces excess mucus. So this affects things like the lungs. You get a lot of mucus built up in the lungs. I think it can also affect the digestive system and the pancreas, but not a fun disease. So let's figure out the chances of having a child that has that. And we'll answer that later, as always. So for the pre-lecture, I'm not going to go through that, but I will do kind of a review here. Not a review necessarily, an overview. So let's talk about genes first. And you can write this down if you want, or you can just listen, or you can skip this if you feel comfortable with all of this already. So a gene is a sequence of DNA that codes for a trait. So I've got a chromosome here, and let's say this little area codes for hair color. So an example of a gene would be a gene for hair color. Next on our list are alleles. You're going to see that word a lot. And these are the specific traits that genes code for. So an example of an allele would be brown hair, or it could be blonde hair. And we assign alleles letters, and the letters are irrelevant. It's just to show which one is dominant or recessive. So with brown hair, it's dominant, so we're going to use a capital letter. And blonde hair is recessive, so we'll use a lowercase letter. And keep in mind, as we go through these examples, there are typically more than one gene that codes for hair color, but we're just simplifying it for you guys. So over here, you get one chromosome from mom and one chromosome from dad. And the gene for hair color is going to be located on the same spot for both. So let's say this chromosome is from mom, this one's from dad, and you got a brown hair allele from mom and a blonde hair allele from dad. So those alleles combine to make a genotype. So an example of all the genotypes that we could get with these two possible alleles are big A, two alleles for brown hair. We can get big A, little a, one of each, an allele for brown hair and for blonde hair, or we can get two alleles for blonde hair. So let's draw our chromosomes over here again with our genes. Again, mom, this one's from dad. And we got a big A from mom and a little a from dad. So the genotype would be big A, little a. Our next word to know is phenotype. And the way that I differentiate between phenotype and genotype is kind of like the genes that you get, and then the phenotype is the physical characteristic that it codes for. And it's not always necessarily a physical trait that you can see, but in the case of hair color, it is. So we use the genotype to determine the phenotype. So some examples of phenotypes with these possible genotypes would be big A, and that would give us brown hair. So the phenotype is the brown hair, and this is the genotype. If we have the genotype of big A, little a, one of each, we're still going to get brown hair because that brown allele is dominant, and so it will overpower the recessive blonde hair allele. And we could get also little a for the genotype, and the phenotype for that would be blonde hair. You have to have two of the recessive alleles for the recessive phenotype to show. So in this case, with the chromosome examples we've been using, we got a brown hair allele from mom and a blonde hair allele from dad. So the genotype, as we determined earlier, will be big A, little a, and the phenotype for that will be brown hair. So hopefully that helps to clarify some of these terms that you're going to see. And let's get started into the packet. So inheritance basics. This is called, I think we talked about this earlier, but a karyotype. And it's when you get a picture of all the chromosomes and you match up the homologous pairs and then you get your picture there. So inheritance basics, humans contain two copies of each chromosome, one from each parent. So for each one, you get one from mom, one from dad, one from mom, one from dad, mom, dad, so forth. So you have 23 pairs of chromosomes. This is the 23rd pair here for 46 total. And each chromosome pair which we call homologous chromosomes, has identical genes. So here we have a pair of homologous chromosomes, one from mom, one from dad. I'm always doing the mom on the left, the dad on the right. That's just how I'm doing it. But anyway, they will have the same gene at the same spot on each homologous pair. For example, the gene for controlling eye color will be at the same spot. And it might not be the same allele at each spot, but that gene for eye color will be at the same location. So that's kind of what it's saying here. Although the two homologous chromosomes have identical genes, what specifically those genes code for are different. So alleles are the specific traits a gene codes for. So an example I've heard before is ice cream flavors. So let's say the gene is ice cream, and it can have different flavors. So you can have an allele for chocolate ice cream, for Rocky Road ice cream, for vanilla ice cream, or the 3-color one, the Neapolitan, whatever. The allele is a specific flavor where the gene is ice cream. So here's a real example. The gene is eye color. And again, please keep in mind that these are simplified. There are multiple genes that code for eye color and hair color, but let's say that the two possible alleles for eye color are blue or brown. Again, those alleles are represented by letters. So we have dominant alleles, and we have recessive alleles. The dominant allele is always a capital letter, represented with a capital letter. Again, the letter itself is irrelevant. Usually we just pick letters that are easy to differentiate between capital and lowercase. So like we wouldn't want to use a C because they look the same in each case. And the recessive allele is always the lowercase letter. So we'll say little a. And the dominant alleles will always be expressed when present, and it will mask the recessive allele if there is one. And recessive alleles are only expressed if you have two copies of the recessive allele, because it's always masked by the dominant allele. So we already had our meiosis lecture, but these alleles segregate during meiosis. So meiosis 2, remember, gives us gametes, which are the eggs and sperm. So you can see here in the father and the mother, these homologous pairs will separate, and you end up with haploid eggs and sperm. Oh, and just to review this picture, if you get a dominant brown allele from dad and a recessive blue allele from mom, you're going to get, the child will have brown eyes because the blue recessive allele is being masked by that dominant allele. So random fertilization. Which sperm fertilizes which egg is completely random, and each sperm and each egg has a unique genetic combination. Remember, we've got crossing over, we have independent assortments, and each fertilization event creates a new being with a unique genetic makeup. And someone did this math, but there is a 1 out of 64 trillion chance of two people having the same genetic combination. So that's not a very good chance, but so you're telling me there's a chance. If anyone knows the movie, you're probably as old as I am. Okay, so gametes with alleles are the eggs and sperm. So here it's kind of hard to see, but this egg has a dominant allele. This one has a recessive allele, and this one has a dominant allele. In the sperm, this one has the dominant allele, this one has the recessive, and the sperm has the recessive allele. And this is coding for skin pigment in this case. So the big A is the allele for normal pigmentation, and the little a is coding for lack of pigmentation, which results in albinism or an albino. So when fertilization occurs here, we get a genotype. This one would be big A, big A. So they would have normal pigmentation. This one, random chance that this sperm fertilizes this egg, but we happen to get two recessive alleles, which will result in albinism, so lack of pigmentation. And these two combined here give us big A, little a. This is going to mask that recessive allele, and we'd still have normal pigmentation. So we combine alleles to make the genotype. And big A we call homozygous dominant. Again, more vocabulary. So homo means the same. So if you see homozygous, the two alleles are going to be the same. Big A, little a is heterozygous. And hetero means, right over here, different, which means you're going to get one of each allele. And then we've got the little a. They're both the same, so it's homozygous. And they're lowercase, so it's homozygous recessive. And like I said earlier, we use the genotype to determine the phenotype, which is the physical description of the genotype. So in this case, whether you have normal pigmentation or albino. So here's another example of genotype determining phenotype, and this doesn't say, but fruit flies are commonly used as examples in genetics. So we can see here there are two different possible phenotypes. There are normal wings, or this one over here is wrinkled. So it looks like having wrinkled wings is recessive. Oh, they're using B here, it doesn't matter. And normal wings are dominant. So this fella here has a genotype of big B, big B. So his phenotype will be normal wings. These two here have a phenotype of big B, little b, big B, little b. So that big B, the allele for normal wings, is going to mask the wrinkled, and those two will have normal wings. But this guy over here has little b, which means he will have wrinkled wings. Now on to Punnett squares. I love Punnett squares. Maybe that's an unpopular opinion, but Punnett squares, we use parents' alleles crossed to give all possible outcomes for offspring, and this will give us genotypic and phenotypic ratios. So in a monohybrid cross, it's gonna be 4 squares like you see here. And this examines the potential outcome of a cross between 2 individuals for a single gene. So mono means 1, so we're just looking at 1 gene. And the different outcomes are represented through a Punnett square, and we're gonna practice these. So the genotypic ratio is the ratio of each of the 3 possible genotypes shown in the results of a test cross. So here's an example down here. What is the phenotypic and genotypic ratio for the following? So the gene we're looking at is the presence or absence of freckles. So having freckles is dominant, it's a big A. And not having freckles is recessive, is a little a. So it wants us to do a test cross of homozygous recessive. So homo means they're going to be the same. Recessive means they're going to be the little a, times heterozygous. Hetero means different, so getting 1 of each allele. And to set up the Punnett square, you have to put all the possible alleles that can be passed on the sides of the boxes. So from this parent, they can either give a big A allele or a little a allele. And from this parent, they can only give little a. And so to find out the different combinations that we can get, you just drag the alleles down and over. So one possible combination is big A, little a. Same here, big A, little a. If we drag these down and over, we get little a, and little a, little a. So the genotypic ratio, we need to look at the homozygous dominant, and there's 0 of those in this Punnett square. We don't have any big A's. For the heterozygous, we have 2. And for the homozygous recessive, we have 2. So this is going to code for, if we look at our phenotype, this will code for freckles. So will this one, because we have that dominant freckle allele. And these 2 will have no freckles, because they got 2 of the recessive alleles. So the phenotypic ratio is 2 to 2, either freckles or no freckles. 2 to 2. I like percentages better. There's a 50% chance of each. So what phenotypes are produced? In this Punnett square, we have 2 with freckles. And 2 without. So basically, if these 2 parents with these genotypes produce offspring, there's a 50% chance that they will have kids with freckles and a 50% chance that they will have kids without freckles. So we're going to practice some more. Complete the following monohybrid crosses as practice. The big A and little a alleles do not have to be freckles. You can come up with your own trait. Let's say this one is for the gene for hitchhiker thumb. And I'm kind of making this up. I don't know what's dominant and recessive here. But this is commonly used in genetic examples. So I have a normal thumb. If I bend my knot like this, it's straight. But a hitchhiker's thumb can be bent almost to 90 degrees. So you can check your own. I've seen some students that are totally like, well. And so let's call the hitchhiker thumb, we're going to say the little a is for hitchhiker's thumb. It's recessive. And the big A would code for a normal thumb. So we've got our Punnett square set up here. Remember, you just drag them down and over. So we had a homozygous dominant parent and a homozygous recessive parent. And so the genotypic ratio, we have 0 that are homozygous dominant. We have 4 that are heterozygous. And we have 0 that are homozygous recessive. No little a's. The phenotypic ratio, 100% of these kids will have normal thumbs. Or you could say 4 to 0. And so what phenotypes are produced in this Punnett square is only normal thumbs. Let's see. Another commonly used example is the ability to roll your tongue. So we'll say that ability is dominant. So we'll call those rollers. Just keep it short. And the recessive allele would be a non-roller. And again, I don't know for real which one's dominant and recessive, but it doesn't matter for these examples. So we've got 2 heterozygous parents. They have one of each type of allele. So in this box, we get big A, big A. Or here we get big A, little a, big A, little a, and little a, little a. So our genotypic ratio would be 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. And when you cross 2 heterozygous parents in a monohybrid cross, the genotypic ratio will always be 1 to 2 to 1. So let's determine the phenotypes here. This one has 2 alleles for being able to roll their tongue, so they're a roller. This one has 1 of each, but the roller allele dominates the non-roller, so this one will be a roller, as well as this one. This one has 2 alleles for not being able to roll, so they'll be a non-roller. So our phenotypic ratio is 3 rollers to 1 non-roller. And again, with a monohybrid cross with 2 heterozygous parents, the phenotypic ratio will always be 3 to 1. So the phenotypes produced are, we got 3 rollers for every 1 non-roller. And I'm going to let you guys pause it here and try this one on your own. You can use any trait you want. You can make one up. But pause it here and fill that in. And I'm going to go ahead and move on. So our question of the day now, what are the chances of having a child with cystic fibrosis? So I'm going to write some stuff out here. It's kind of a lot, so bear with me. Cystic fibrosis, sometimes called CF, is a recessive disorder meaning there must be 2 copies of the defective recessive allele for the individual to have it. So it also depends on the genotypes of the parents. So in this example, we have two heterozygous parents. So they are carrying the allele for cystic fibrosis, but they do not have cystic fibrosis. And they may not even know that they're both carriers of cystic fibrosis. So a big B is going to be the normal allele. So someone without cystic fibrosis or not coding for cystic fibrosis. And little b is the allele for cystic fibrosis. So what parental genotypes would lead to the disorder? Well, if you have two parents with cystic fibrosis, they're both going to be little b. And so if we have little b here, little b here, little b here, and little b here, 100% of their offspring will have cystic fibrosis. So two individuals with cystic fibrosis, if they have kids, all of them will have cystic fibrosis. But in this case, we're looking at two heterozygous parents. So we've got big B, little b from mom, big B, little b from dad. We drag those down and over. So any genotype with a big B, since it's dominant, will not have cystic fibrosis. So these three would not have it, but this one here would have cystic fibrosis. So in a heterozygous parent, there is a 1/4 or 25% chance of a child with CF. And something to remember, let's say they have one child and they have cystic fibrosis. That doesn't mean they can have three more and guaranteed not have cystic fibrosis. Like this resets with each new offspring. So every single time that they have a child, there's going to be a 25% chance that that child will have cystic fibrosis. So what parental genotypes could lead to the disorder? Big B, little b, two heterozygous, or like I said before, if both of the parents have cystic fibrosis, that will lead to the disorder as well. So if you thought it couldn't get any more complicated, we're going to do a dihybrid cross. Di means two. We were previously doing monohybrid crosses with just looking at one gene, but this examines potential outcome of a cross between two individuals for two different genes. So we're going to look at the gene for freckles, whether freckles are present or not, and then short fingers versus long fingers. So according to this key, freckles are dominant to not having freckles, and having short fingers are dominant to having long fingers. So in meiosis 1, we have our pair of homologous chromosomes, and those are separated in meiosis 1. And it's showing the genes here. My picture is kind of too blurry to point out, but hopefully you can see it on yours. And then in meiosis 2, we separate those sister chromatids to give us haploid cells. You can see we have lots of different possible combinations of those genes or alleles in the eggs and the sperm. So let's do our dihybrid cross here. And this one we're going to have 16 squares. It's a 4x4. So remember, finger length is represented with the letter A. Freckle gene is represented with the letter B. Big A, short fingers are dominant to little a, long fingers. And big B, freckles are dominant to little b, no freckles. So what is the phenotypic ratio of a cross between two heterozygous for both freckles and finger length? So first, let's determine the genotypes of the parents. They're both heterozygous. So for finger length, we would have big A, little a. And then for freckles, we would have big B, little b in both parents. And now we just need to determine the different combinations that we can get for each gene for each parent. So for this first one here, they could give a big A and a big B. Or they could give a big A and a little b. Or they could give a little a and a big B. Or a little a and a little b. And since both our parents have the same genotype, we can copy the same thing down the other side for the other parent. And then we drag them down and over, and we keep the same genes together. So this one's big A, big A, big B, big B. This one would be big A, big B, little b. And it's just formatting. We always put the dominant allele first. So big A, little a, big B, big B, big A, little a, big B, little b. On to the next row, big A, big B, little b, big A, little b, big A, little a, big B, little b, big A, little a, little b, little b. Here we could get, I'll stop talking, just watch. I'm saying too many bigs and littles and A's and B's. Okay, so it asks us our genotypic ratio. I'm actually going to write out the genotypes. One possibility I see is big A, big B, big B. So let's see how many of those we have here. Just one. Another one I see is big A, big B, little b. And we have one, two, just two of those. Another possibility I see is this one. Big A, big B, big B. We've got one, two of those. We can also have heterozygous for each, so big A, big B, little b. And I see one, two, three, four of those. And then I see this one here, big A, little b, little b. One, two. And then we can have little a, big B, little b. We've got two of those. And then last one, little a, little b, little b. So that's kind of a painstaking process, but our genotypic ratio, and it's always this way in a dihybrid cross, if you cross two heterozygous parents, will be 1 to 2 to 2 to 4 to 2 to 2 to 1. So let's write out our phenotypes for each one. This one will have short fingers and freckles. I'm just going to say yes for freckles or no. This one will have short fingers and freckles. This one will have short fingers and freckles. And this one will have short fingers and freckles. This one will have short fingers and freckles. Short fingers and no freckles. So we've got two homozygous recessive. This one will have short fingers and yes, freckles. Short fingers and no freckles. Short fingers and freckles. Short with freckles. We've got a long finger finally with freckles. And we've got another long with freckles. And we've got short with freckles. Short with no freckles. Long fingers with freckles. And long fingers, no freckles. Hopefully you're still with me here. So the phenotypic ratio, one possibility is short fingers with freckles. So let's count those. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 there. And then we can have short fingers with no freckles. So if we count those, we've got 1, 2, 3. That's 3. And we can have long fingers with freckles. So we've got 1, 2, 3 of those. And we can have, long fingers and no freckles. So our only long and no is this one. So the phenotypic ratio in a dihybrid cross with heterozygous parents will always be 9:3:3:1. And what phenotypes are produced, we can have basically all the combinations. Short fingers with freckles. We can have short fingers, no freckles. We can have long fingers with freckles and long fingers with no freckles. Okay, and I know this page was a lot, and I believe there is a question on the exam about a dihybrid cross, so this is something you'll probably want to practice. And I believe on Canvas, there are a couple of documents attached where you can practice, and then there's an answer key. So I would recommend going through that and message me on Canvas if you have any questions. Bye-bye.